covering and what I'll be covering are kind of complementary, and I wanted to bring that up with this picture. Although, what you see on the picture is kind of specific towards ecological systems, but you can pretty much replace those pictures with pictures more relevant to what Pranay is talking about, and the story would still go through. So what is this picture saying? Essentially, when you are dealing with any biosystem, in this particular picture we are talking about essentially ecological systems. So you have essentially a predator trying to catch a prey, presumably to eat it and increase its numbers. You could try to go in two directions. One is you could try to understand all the delicate nuances of the dynamics underlying this, whatever is going on here. So, most of you might have heard of the Lotka-Volterra system of equations. So that's in some sense the simplest mathematical description of how a predator and a prey's population affect each other. So you could try describing it with such a simple two-variable coupled differential equation, but then you encounter reality and you find that the actual variation across time of the populations of a predator and a prey species looks somewhat like this, very far removed from the extremely periodic looking time series that you generate from the Lotka-Volterra equations. So at that point, you have two choices. You could try to build in more details. So for example, you can build in what is known as functional response, or you could build an age structure. So some of this would be dealt with Malay when he covers ecological modeling next week. Or you could say that, well, in reality, these two species are not isolated. They're actually part of a much larger ecosystem. Okay? There are, of course, abiotic factors. So, you have changes in rainfall, changes in other climatic patterns, and so on, which affects their numbers, apart from, of course, their interactions on each other. But they're also part of this entire food web in which this particular prey species itself feeds on other things. So, from that perspective, this becomes the predator and something else becomes the prey. And in turn, the predator over here could itself be the prey of something else. And you could ask that, could it be that the deviation that you see of the time evolution of the populations of these two species is because we are looking at them in isolation and not considering them to be connected to a much larger number of species whose interactions in turn affect the population variations of these two species. So at that point, you start getting interested in how different species relate to each other in terms of predator-prey interactions, or could be just competitive interactions. So they could be interacting with each other not because one eats the other, but simply because they both feed on the same type of food, and so therefore they are competing with each other. So you could have an extremely complicated looking network, and I use the word for the first time now. I'll explain it, what it means much in a detail immediately afterwards. So you can actually construct such a network picture of how the species interact with each other. And then you could try to bring in the dynamics that you were studying in this axis into the mix. And then you can study the dynamics on complex network, which in some sense is a very exciting area to work on, where you can try to understand how do extinction cascades happen. So how is it that the drop in numbers of one species result in a steep drop in the number of other species, which in turn will lead to steep drop in other species and eventually would lead to ecological collapse. So although these two axes may look orthogonal. There is, like when you are dealing with, extremely complicated model of two species interactions, you don't necessarily have to know what kind of food web they are part of. And similarly, when you're, constructing food webs, you don't necessarily have to understand any of the dynamics which goes on in predator-prey interactions or two species competitive interactions. It's only by an appreciation of both of these axes that you finally get to understand real phenomena like ecological collapse. Okay. So I use the example of ecological systems, but now you can actually replace the dynamics with whatever that you are specifically interested in. So for example, to take Pranay's example, I can now replace it with two neurons. And I could, for example, if I go along this axis, be extremely interested to understand how does, let's say, two excited neurons which are interacting with each other synaptically result in certain kinds of firing patterns. And at the same time, I might be interested to know how is it that neurons in a particular nervous system or even a particular region of the brain are connected to all the multifarious other neurons that this particular system is comprised. Okay. So if I'm essentially constructing such a brain network, I may be completely oblivious of the specific dynamics of these individual neurons that are there. But then eventually at some point I would bring in Pranay's models, whether it be Hodgkin-Huxley or whether it be something far more complicated, and I would bring in the network of neurons or network of brain regions that are constructed from one of a number of techniques that at some point in the lecture series I would cover. And at that point I can study the dynamics of the brain as a whole. How does the collective dynamics of neurons give rise to perception or give rise to the other kinds of phenomena that we associate with conscious beings? Right? So it's often said mathematics is a language to express nature. Okay? So given that both of these axes are different manifestations of mathematics, you can well ask, so then if mathematics is a language, what are, let's say, dynamics and what are interconnections or what is study of networks? I like to think of them as, well, one could say different types of language. I don't know whether you've heard of the early 20th century linguist called Benjamin Lee Whorf. He had kind of a structuralist view of language and how it affects cognitive skills of human beings. So the Whorfian hypothesis was that the kind of language that a person uses essentially colors or even constructs the worldview that they have. So he had, in fact, a cartoon in his book, Language, Thought, and Reality, where he said that when a French person speaks about a particular scenery, the worldview that this person has would be very different from when a person who speaks English takes in the same scene and tries to describe it. And one of the examples he gave to point this out was that when we talk about snow, or when he talks about snow, there's only so many adjectives that he could use to describe snow. However, when an Eskimo describes snow, there would be 9 or 10 different ways that the Eskimo would classify as snow. So the very setting in which a person comes from, the very setting whose language that the person uses, essentially describes the perspective that this person is going to use to describe the phenomena. And in a sense, networks are a perspective. It's a way to view any phenomena, which will be quite different and sometimes complementary to the other kinds of phenomena that you might use. Okay, so now we come down to brass tacks and we define what is it actually that we are talking about. What is a network? Okay. So in the simplest terms, a network is described by two things. Okay? It's made out of a set of nodes or vertices and the edges or links which connects these nodes or vertices. And so depending upon what system you're interested in, these nodes and these links could have different interpretations. So if you're a neuroscientist, your nodes would be neurons and your links would be synapses or electrical gap junctions which allow the neurons to communicate with each other. On the other hand, if you are a cell biologist, your nodes could be proteins and your links could be the interaction, the physical binding of these proteins with each other. If you're an ecologist, these nodes would be species and the links would be the trophic relations which define which species eats which species competes with which species and so on. On the other hand, if you are an epidemiologist interested in how infectious diseases spread through populations, these nodes would be individuals and the links would be the physical connections, the physical interactions through which the contagion jumps from one individual to another. So you can see it's a very general framework. You can just take your favorite system, and it's actually a very good, homework for you that, you can just think of some system that you're interested in and try to translate into a network language. So you would have to first identify what are the nodes in such a description and what would correspond to the links in such a description. And so if you say that the set of nodes correspond to the set V and the set of all the links correspond to E, so the combination V comma E would essentially define the graph or the network, which essentially corresponds to the system you're interested in. And you could, of course, graphically represent the networks like this. So you could represent the nodes as circles and all the links that occur between them as lines like this. But of course, when you are dealing with them mathematically, it's kind of cumbersome to essentially represent every network that you have in such a graphical representation. So instead, we would represent it using a matrix, which for various reasons is called an adjacency matrix. Why adjacency? Because it tells you which node is adjacent or is connected via link with another node. And so if you have a n by n matrix, essentially it means that it tells about how the n nodes that make up that network are connected to each other. So if you have the ijth element equal 1, that means that there is a link which connects node I to node j. And if it's 0, then there's no such link. Now, this is the very basic definition, and then now we can build in more complexity. So you can say that, well, here I have assumed that if a connection exists from I to j, then a connection also exists from j to I. Okay. So this is what is known as an undirected network. So the link is essentially not a property of, does not have the property of directionality. So it just exists. On the other hand, if you're thinking of something like a synapse, if a particular neuron I has a synaptic connection to a neuron J, that always has a direction, right? Because you talk about a presynaptic neuron and a postsynaptic neuron. And just because neuron I has a connection to neuron J does not necessarily mean that J would also have a synaptic connection to neuron I. And so there we define what is known as a directed network. And so there in general, you would have an asymmetric adjacency matrix. So Aij does not imply that you know what is Aj. So Aij not equal to Aji. And you could actually then build in further complexity and start talking about weighted networks, where you say that, okay, adjacency matrix only tells me whether a link exists or not, but all links may not be equally important. Okay? So think of a social network. Okay? You may in the course of course of 24 hours, have conversations with lots of people, but some of these conversations would be like 1-minute conversation. Other conversations could be much more deep, right? So you could have like a half-an-hour conversation with a friend, and you could have like a 30-second conversation with someone from whom you're buying a ticket. Okay? And if you want to quantify this difference in the nature of the interactions, you have to associate a weight with these links. And so then we start talking about weight matrices for the connections and so on. Okay? But these are all further details. You can now start thinking of how to bring in more and more features of actual reality into your network description. But we start essentially from this simple description of a network, that of an adjacency matrix, and moreover an undirected network. So essentially you are talking about a symmetric adjacency matrix. Okay, so that essentially answers the question, what are networks? But you might be asking, why networks? Why is it that networks are at all interesting, or we should be looking at networks? Because, at least that's my answer to it, because I believe that networks provide one of the more comprehensive answers to the question about how do we get a system of components to behave very differently from the behavior we associate with the individual components. A more succinct way of putting this is to say more is different, which is in fact the title of an article that the physicist Phil Anderson wrote in Science back in 1972, way before all this network craze. And so what he pointed out in this article was that if you look at different sciences, you often find that there exists a pair of sciences where science X essentially describes the behavior, or rather the other way around, the science Y describes the behavior of the components of science X, but it's not that science X is just applied science Y. Okay? And essentially he pointed out that if you look at solid state physics, essentially you're talking about the interaction between electrons, crystal lattices, and so on. So all of these particles are of course following the laws which are given by elementary particle physics. Okay? But at the level of solid state physics, you have lots of properties arising like band structure, superconductivity, and so on, which not necessarily you can derive just from knowing the principles of particle physics. These are genuinely novel phenomena which only emerge at the scale of million or billion of these particles interacting with each other. Similarly, when you look at chemistry, we are talking about interaction between molecules, and molecules are of course following the principles of many-body physics, but just using many-body physics, you cannot often figure out how, for example, when you make an acid react with some substances, what's going to happen. And he goes all the way from, let's say, chemistry being the fundamental bedrock on which molecular biology is embedded, and molecular biology being the fundamental bedrock on which cell biology is embedded, all the way to physiology essentially forming or underpinning the laws of psychology, and psychology in turn essentially being the bedrock on which social sciences are set up. So essentially what he meant by this was that if you look at a crowd is of course made up of individuals, but knowing each of these individuals would often not be enough to understand how the crowd will behave. And we all know of this example where a crowd made out of completely rational individuals who in isolation will shudder at the thought of violence, mindless violence. Nevertheless, when they're put in a large enough crowd, will indulge in looting, arson, and worse. Right? So this has often been commented upon by social scientists, about the crowd mind and so on. But Phil Anderson was seeing this as an example of emergence at a systems level of properties which are absent in their components. Okay? And so he put this very succinctly as follows, that just because you have this hierarchy does not imply that the science X is the applied form of science Y, because whenever you go from Y to X, you have the emergence of completely new set of phenomena which you have no way of explaining or even understanding from just pursuing the laws which define science Y. Okay. Now this may look abstract, so I will take an example of a science I know particularly well and tell you how this comes about. But just to reiterate, what Phil Anderson was talking about is what we would nowadays call emergence, and in fact that's something Pranay mentioned in his talk. The emergence of behavior qualitatively different from the individual components in a network of interacting components. And if you think of an example, let me take Pranay, the stuff that Pranay is talking about. So the components could be neuron. The interactions could be synaptic interaction, gap junction interaction, and so on. And the system is the brain. Now, of course, it's given that neurons are quite complicated objects. But however complicated they are, I think all of you would agree that once we put 10 to the power 11 of these neurons together in a particular configuration, and you get this system, which effectively now is giving this talk to you, I think all of you would agree that something new has occurred, because an individual neuron, however complicated it is, will never have the capacity to essentially be capable of the kind of behavior that the overall brain is capable of. And so networks, or the network science, essentially tries to understand how is it that interactions result in this novel phenomena, what we would call the complexity at the systems level. So let me take an example of a subject which presumably all of you have studied at high school, if not at a higher level. So the question is, why do we need this science called statistical mechanics or statistical physics? Okay. What is it actually doing? Okay, so before statistical mechanics came along, you had two very separate bodies of science which dealt with the science of a few bodies interacting with each other and a science of large number of particles interacting with each other. So at one end, you had classical mechanics, which essentially dealt with the behavior of a few particles. And essentially, they were described by just the position and the momentum of the individual particles. So they were described phenomenologically way back in the 17th century by Kepler. And then we had the solid foundation laid by Newton. And so once you have the Newtonian equations, you think that you have a very comprehensive body of knowledge which describes how a few particles interact with each other. Now, parallel to the development of classical mechanics, you had thermodynamics, which essentially dealt with a huge number of particles, essentially an Avogadro number of particles, so 10 to the power 23 particles. And because position and momentum didn't make sense when you're talking about such a huge astronomical number of particles, you had completely different variables which described them. So you had pressure, you had volume, you had temperature, and people like Boyle and so on essentially came up with laws which related these variables to each other. Just as Kepler came up with phenomenological relations which essentially related the planetary motion, similarly Boyle, Gay-Lucas, Charles, so on, came up with phenomenological laws which related the pressure, volume, and temperature of a gas. And in turn, these were given solid foundations by the work of Carnot, Clausius, and others, and you had the laws of thermodynamics. So on the one hand, you had Newton's laws, which described the behavior of a few particles. On the other, you had the laws of thermodynamics, which told us how a large collection of particles behaved. Now, where the puzzle lay was that you had phenomena occurring at the scale of thermodynamics, which simply had no analog at the scale of Newtonian mechanics. Case in point, the arrow of time. Classical mechanics is completely time reversible. Okay? So if you look at the motion of a single particle, if you look at the laws, if you look at Newton's equations, if you replace t with minus t, makes absolutely no difference. And if you show me a film of a classical particle essentially moving about in a dissipation-less environment, there's no way I would be able to tell whether you're showing me the movie forwards or backwards. Yet when you come to the domain of thermodynamics, it's very easy to say which way a film is going, because I can take a glass, I can release it, it will fall on the ground and shatter to pieces. So if you now show me the film backward, so that suddenly there's a whole lot of glass shards on this floor, and then suddenly all of them decide to come together spontaneously, form a glass, and then jump up the floor and reach my hand. Any of you would say that it doesn't happen, you're just showing a movie backwards. So how do we know that there is a directionality of time? Because certainly Newton's laws, which govern each of those particles which make up this glass, follows a time reversible set of equations. So where is this arrow of time coming from? Where is the irreversibility coming from? Okay? And it took the genius of Ludwig Boltzmann to formulate statistical mechanics, which gives us the answer that how is it that from reversible laws of governing the motion of individual particles, you have essentially the irreversibility that we associate with thermodynamics. Okay? So essentially what we are looking at is a kind of statistical mechanics, but not something which just applies to inert particles of large volumes of gases, but which can apply to anything. Okay? And so I like to think of networks as network science as a generalization of statistical mechanics to all kinds of entities. Okay? So essentially, you have the situation that now we are dealing with not just particles of gas, but it could be neurons, it could be species, it could be human individuals. And the question we're asking is, when they interact with each other, how is it that even though we completely, let's say, understand the principles govern the behavior of each individual particle, as a collective, how is it that new phenomena can emerge? And of course, our endeavor is further complicated by the fact that unlike the physics example I gave, typically these systems are often far from equilibrium. They're driven. Okay? And so we have to bring in new concepts, new principles. So that's why we can't just use statistical physics as investigated by physicists over the last century or so and just apply it mindlessly to understand all these new systems, biological inspired systems that we are looking at, but we need essentially new principles. So the question that we are asking here is that, are there universal organizing principles behind biological phenomena that occur at very different scales? So just to give one example, we can ask that you have signaling machinery which acts as the nervous system when you are dealing with at the level of the brain, but you also have very similar signaling networks which occur at the level of a single cell. The components are very different. Okay? In the case of the cell, the role of the neuron is taken by molecules, and the interaction could be chemical reactions. On the other hand, in the case of neurons, of course, the components are not molecules, but cells, and the interactions are synapses. But what about the principles which operates? Could the principles be very similar? And in fact, one question would be asking is, why is it that nature chose networks rather than dedicated pathways? Why is it that when you are designing something like the brain, you just don't have a collection of reflex arcs where a particular sensory neuron is dedicated for a particular purpose, and that in turn connects to a particular dedicated motor neuron. And for each possible input-output response, you have separate such reflex arcs. Nature could have used that, but it didn't choose to do that. Okay? It decided to connect all these neurons together in a very complex network. There is, of course, a price to pay for this. So why did nature choose to pay this price rather than come up with this very simple set of separate reflex arcs? Now, you might think that's a question very specific to nervous systems. But then when we come to intracellular signaling, we find that, again, nature chose precisely that. So it could have come up with just separate pathways. Okay? So you have very specific receptors located on the cell membrane, and you could have dedicated molecules which only respond to that particular receptor, and eventually you would have a certain kind of pathway which is triggered by a very specific signaling molecule, which are all separate from each other. That, in fact, was a cartoonish picture which people believed till about 5 decades back, but now we know that the cell signaling pathways are actually all connected with each other. It's actually a network rather than a set of pathways. So why is it that at a completely different scale, again, nature chose to go with a network rather than a set of pathways? Are there certain advantages to be had in signaling when we are taking the network approach rather than the separate pathways approach? So these are the kinds of questions that transcend levels, that transcend systems in biology, and are best addressed by using the network approach. So in this first lecture, what I thought I would do is I would introduce you to the different scales at which you can apply the network perspective in biology. And I would essentially take examples from each of these levels interchangeably in the next set of lectures. So I thought I'd first introduce you to the specific systems that I'll be dealing with. And while we are at it, I will try to introduce very briefly the basic concepts that I'll deal with in greater detail in the next 5 lectures. So the one thing that strikes us when we look at biology from a network perspective is that networks seem to be there at almost all scales, right from the scale of molecules to the largest scale possible in the biological context, namely that of ecologies, planetary-wide networks. Okay? So at the smallest scale, you can think of a protein itself as a network, as I will describe in detail in the next few slides. If you go up little, you have a large number of networks which occur inside the cell. So you could have the intracellular signaling network, you could have the metabolic network, you could have the network of protein-protein interaction, and so on. At an even higher scale, you could have networks where the components are cells, and so you have the brain as a prime example of such a network. At an even higher scale, the nodes could be individuals, and the interactions could be social interactions. And the kinds of phenomena that are of interest at that point are epidemics, something that Gautam would be dealing with in greater detail. And at an even higher scale, you would have species which are interacting with each other in trophic relations, and you have food webs. Okay? And so these are just a few examples of the huge, incredible number of networks that we come across when we look at biology from a network perspective. Okay, so let's start right at the bottom. Okay? So those of you who have biology background, or those who have studied proteins before would of course know this very well. But in case you have not encountered, if you are coming from a different background, essentially a protein or any protein is made out of linked amino acids, which in normal circumstances, if you put it in its right environment, occurs in a folded form. Okay? So you can take a protein, you can force it to open up, but if you let it go in its proper environment, it will always fold into a unique configuration. Okay? And it's important that it always folds into the unique configuration because the structure that it takes, the structure that it adopts, is actually important for its function. Okay? Because many of the reactions that proteins are involved in occur by what are known as lock and key mechanism. Okay? So essentially a protein when folded would have a particular conformation, and it tries to find a binding partner, and when it finds a binding partner which just exactly fits the contours that are made by this three-dimensional configuration, a particular reaction happens. Okay? So we can view the three-dimensional structure of the protein essentially as a network where the nodes are the residues or the amino acids that make up this protein. So here they are represented by these small spheres, and the interactions correspond to the non-covalent interaction between the residues. Okay. Let's take an example. So Pranay has already introduced cell membrane, and he has talked about ion channels. So let me take an example of a specific ion channel. So this particular potassium ion channel, people have essentially found its structure. So you can find that, you can see that it's represented very stylistically as a set of helices and beta sheets. So these are structures, or the so-called secondary structures made by the linked amino acids, which when you let them evolve in their natural circumstances would spontaneously form these helices and sheet-like structures and so on. So it looks very complicated, but you can actually try to view it as a network as follows. So you can essentially replace each of the residues by just a node, and each of these nodes, as you can see, are linked with each other through chemical interactions. Now, when you look at it from a network point of view, you realize that it has a certain kind of conformation that is quite unique for it. And you would like to understand this in a bit more detail. So what do? You want to first translate this into an adjacency matrix. So how do you translate this into an adjacency matrix? So one way to do that is to obtain the structural data corresponding to this protein. So for example, you can go to protein database, PDB. You can give the identifier for this particular protein, and you would get essentially a table which tells you what are the three-dimensional coordinates of each of the residues which make up the particular protein we're interested in. So for example, here we are looking at this ion channel, Kerbach 1.1, and essentially these three columns corresponds to the x, y, and z coordinates of each of the nodes which make up the protein. Okay? I'm just only showing you the first 38. It will of course just continue. Now what you can do is you can ask, what is the distance between every pair of residues? In this particular case, I'm just asking, what is the distance between every pair of the carbon alpha atoms which is part of each residue? Okay. I calculate the Euclidean distance between every pair, and I can represent it as a matrix. So the color code for this essentially represents in terms of angstroms, what is the distance between every pair of residues. So you take any entry of this matrix, read off the value associated with the color, and it will tell you how close two atoms are in that particular protein. Okay. So you would realize immediately that there is something like a repetitive structure or 4 repeating motifs in this particular matrix. So that in fact is related to the fact that the ion channel protein is in fact made out of 4 identical subunits, which make out the petals of a flower, so to say. Now, next what you would like to do is you want to just focus on the relevant distances, the distances relevant for the interactions you're interested in. So I mentioned that here I'm interested in the non-covalent interactions between the different atoms. So I look at my chemistry book and I say that, look, if 2 atoms are beyond 12 angstrom, it's very unlikely that there would be any van der Waals interactions between them. So I can safely use a cutoff of about 12 angstrom. So I say that every distance which is greater than 12 angstrom, I say that a link does not exist, and the rest I say that a link exists, and so therefore I just translate distances above 12 angstrom to 0 and distances less than 12 angstrom to 1, and I get a matrix which consists of just zeros and ones. So it's just an adjacency matrix. Now you notice that it retains exactly the repetitive structure of the original distance matrix. So you can magnify each of these repeating structures, and you find that each of those elements themselves have what we call a modular structure. In other words, if you look at the matrix, you find that it has a block diagonal structure. That is, it consists of 2 blocks where connections only exist within each block. There are almost no connections between these 2 blocks. And if you now try to associate with the original protein, you find that this essentially consists for the membrane domain, and this corresponds to the intracellular domain of the protein. So if you go back and look at the representation, the visual representation of the protein embedded in the membrane, essentially you would have one part which is embedded within the membrane, the other part which is free and in the intracellular medium. So essentially this is the part that corresponds to one of the modules, and this is the part which corresponds to the other module. So you can see that even at very superficial kind of analysis, your adjacency matrix captures some of the information that you would have gleaned from a more detailed perusal of the structure. Now, you can also be interested in the so-called degree distributions. So what is degree? So degree is the name that we associate with the number of connections that each node has. To use a social analogy, if you say that degree corresponds to, our social degree corresponds to how many friends we have or how many acquaintances we have, we can distinguish different individuals in terms of how large is their acquaintance set. So a high degree node in such a setting would correspond to someone who's very popular, who has lots of acquaintances, and someone who's a loner, who probably only talks to one person, would be a very low degree node. So we can look at the degree distribution of the different residues for this particular protein and find that there would be a few nodes which would have roughly about 60 other residues within 12 angstrom of it. And so therefore, in principle, it could be having van der Waals interactions with lots of different residues. There would be also quite a few which would have very few such interactions. Okay? So the ones which belong to this end. And often, just by looking at which nodes have high degree, you can identify functionally important residues. Okay? you can try to bring in more physical insight into this network picture by now bringing in some high school mathematics. So you could say that, look, I can try to say something about how this protein behaves by viewing it as an elastic network where essentially the residues correspond to particles or balls, and the chemical interaction essentially correspond to springs. Okay? So now, for anyone who has, done preparations for joint entrance examination, you'd remember that you were told to analyze systems of particles connected by springs or systems of masses connected by springs. So just increase the complexity of the problems you were dealing with. You were maybe dealing with, let's say, 3 particles connected by 2 springs. Now just imagine there are n particles which are connected by any number of springs. Okay? So you have essentially a system which looks like this. Okay? So now how would you analyze such a system? Well, you can try to write down the potential energy function for this system. And we use what is known as the harmonic potential approximation. So we essentially say that, look, every particle has an equilibrium position which is given by all the other particles that it's connected to with springs. And if you displace the particle from its equilibrium, essentially it would come back after a bit of time because the energy would always try to go to its minimum. So how do we write down the energy function? Under harmonic potential approximation, we can essentially expand the energy of any displacement of an amino acid from its equilibrium position as a power series expansion. Okay? And we can assume that we are only looking at small deviations about this equilibrium position. So we cut off all higher order terms over the quadratic term. And since at equilibrium, the first derivative of the potential energy function with respect to the displacement is simply 0, that's the definition of an equilibrium. Essentially, we are just dealing with the energy at the equilibrium and the second order term. And you can redefine the origin of your energy function to essentially make this 0. So finally, we are just left with the quadratic term. So this is known as the harmonic approximation, okay? Harmonic potential approximation. So for such a network, you can now write down the energy function of the entire system as simply now a sum of the squares of all the deviations of the distances of pairs of residues from their equilibrium position. Okay. As I said, all you have to do is think about what you did, or what the kind of practice you did during a joint inference, and instead of just looking at 2 or 3 particles connected by springs, you just translate it to n particles. Okay? And through the use of a potential energy, you can simply write down the energy corresponding to the network of n particles connected by springs as simply a sum of the squares of the deviations, instantaneous deviations of the particle positions from their equilibrium position. Okay. Now, just by using vectors, I can work out that this essentially translates to nothing other than the square of the difference of the instantaneous deviations of the ith residue and the jth residue. Because if you look at what is happening here, so I'm talking about, over here I'm talking about the deviation of the equilibrium distance between residues I and j, rij naught, from the instantaneous deviation rij. Now, just by using vectors, I can associate it with simply the delta Ri, which is the deviation of the ith residue from its equilibrium position to its current position, and the delta Rj, which is the deviation of the jth residue from its equilibrium position to its current position. Okay. In case any of you are kind of wondering how is it that I can do this, it's very simple actually. So all you have to do is notice that R I naught and delta Ri are simply related, are simply the arms of a particular triangle, and similarly for R j naught and delta Rj, and you can just work out through simple vector algebra the fact that Rij minus Rij naught is nothing other than delta Ri minus delta Rj. In fact, it's worked out over here. So once I do that, once I have the energy function for the entire system, what else can I do? Now, I could try to solve this. I could try to solve how small fluctuations would eventually go away by treating it as a set of coupled differential equations using Newton's laws. But instead, I will do something else. I will translate this into a matrix form. Okay? So what I'm doing over here is that instead of thinking of it as a sum of several entries, I'm just writing it in a matrix form where each of the deviations, delta R1, delta R2, delta R3, are essentially written as a column vector of deviations and a row vector of deviations, mainly it's transpose. And I can view this operation as nothing other than a column vector multiplied by a matrix and operated upon by a row vector. And of course, you have this stiffness constant which was there before. So all I have done is I've translated something which is written as a sum of separate terms into a matrix form. Okay? But in doing that, I've essentially introduced a matrix L. Now, what is this matrix? So it turns out that this matrix is so important and occurs in so many different contexts, it has a name of its own. Network scientists typically refer to it as a Laplacian. In the very specific context of protein contact network dynamics, it's also sometimes known as the Kirchhoff matrix. Okay? And if you just try to associate this particular matrix expression with this original sum of several terms, you'd quickly realize that for this to reduce to this, the matrix L has to have the following properties. All the off-diagonal elements which correspond to two residues having a connection would have the entry -1, and it's 0 otherwise. And all the diagonal elements equal the degree of that node or the sum of all the links for that node. Now, just try to recall for a moment the definition of the adjacency matrix. What was the adjacency matrix? So it was defined as follows. Aij equals 1 if I connected to j and equal to 0 otherwise. Okay. And now here you have the definition of L. Do you see the connection between the adjacency matrix and the Laplacian? How will we get the Laplacian from the adjacency matrix? So if you know the adjacency matrix, is there some way we can construct the Laplacian? Or is it that you need some additional information? That's a question. Anyone? Sorry? No, far simpler. No power is necessary. It's far simpler. Just look at the definition of the Laplacian. So what's the definition of Laplacian? So the Laplacian is basically -1 if ij are connected and equal to 0 otherwise, Okay. And the other thing is that Lii is simply equal to, let's call k the degree, so k of I. Okay. So now just think, what is the relation between A and L? The off-diagonal element is easy, right? It's just the negative of the corresponding Ai, J. What about this one? It's the number of connections. Now, knowing adjacency matrix, can I say what is the number of connections of a particular node? Exactly, and for a symmetric matrix, it doesn't matter whether you take a sum over rows or sum over columns, right? And so this tells you that, wait a minute, if I just write it as summation Aij, let's say I sum over j minus Aij well, I mean, this, of course, I put a canonical delta function. Okay. All right. So, so basically, this tells you that knowing the adjacency matrix tells me the Laplacian. And once I have the Laplacian, I can actually talk about the correlations between the fluctuations, where essentially I just bring in this Boltzmann factor into temperature, because I'm talking about thermal fluctuations, and the K is just the stiffness constant over here. But the important part is, here I have this reciprocal of the Laplacian. And so this tells me that if I'm looking at the small elements of the Laplacian, or rather the smallest eigenvalues of the Laplacian, they would correspond to large fluctuations. And so by now focusing on the eigenvalues of the Laplacian, by looking at the diagonalized matrix, the Laplacian, I can actually look at its eigenvalues, and the small eigenvalues which tell me about the large scale motion of the protein, whereas the larger eigenvalues tells me about the small scale motions. And so now I can actually look at the eigenvalues, or the magnitude of the eigenvalues. So here I've represented the reciprocal of the eigenvalues, because essentially I'm interested in the smallest eigenvalue. So if I look at the reciprocal, then the largest values of the reciprocal eigenvalue would correspond to the smallest eigenvalue. So, and you notice that there are 4 eigenvalues which are essentially separated from the bulk. Okay. Notice that this is in log scale. So these 4 values are actually much larger than those in this bulk. So what this tells me is that there are 4 normal modes which are actually very large compared to all the other vibrational modes that we associate with this protein. So now what we can do is we can look at the eigenvectors associated with these modes and try to understand what are the components, what are the residues which are associated with these modes of motion. And we find that in fact there is a repetitive nature of this eigenvector. So it looks a bit busy, but essentially the black curve corresponds to the eigenvector components of the second eigenmode. The red curve corresponds to the eigenvector components of the third eigenmode, and the blue curve corresponds to the eigenvector components of the fourth eigenmode. So you realize that there is a repetitive structure which corresponds to these 4 identical subunits, but not all the components of all the subunits take part equally in each of these vibrational modes. So if you want to understand this in a bit more detail, we can actually construct a graphical representation of this information. So what I've done over here is I've shown the second, third, and the fourth components of these second, third, and fourth eigenmodes, and I've portrayed that in a 3-dimensional landscape. Now you notice that some of them are colored red and some of them are colored yellow. And so what I've done over here is that I have taken those residues which correspond to the intracellular domain and I've painted them red, and I've taken those residues which are part of the membrane domain and painted them yellow. And this tells me that the one which is in the intracellular domain, those 4 subunits are essentially, unfolding in a particular manner, and they are not, essentially in consonance with each other, while the part which is in the membrane domain is much more rigidly fixed. And we get an understanding of the overall large-scale opening and closing motion of the protein simply by looking at the structural data. So all you had started with was basically the adjacency matrix, the structured data. You just did some analysis on its Laplacian, and out comes an understanding of how the protein as a whole opens and closes as a result of these fluctuations. Okay? So this is, in some sense, just an inkling of often the kind of insights that even a very simple-minded network analysis can give about the underlying dynamics and, by extension, the function of the system you're interested in. Incidentally, a very similar analysis for the internet, which can also be viewed as a network, was done way back in 2003, which essentially showed that just by looking at the overall backbone structure of the internet and analyzing its Laplacian precisely in this manner and unfolding it into the space given by the largest few eigenmodes, you can see that essentially the internet is not like a homogeneous connected network of servers, but has very specific compartments located at specific continents. So there are quite a set of insights that one can glean from even the most rudimentary network analysis. And I mentioned degree. So the degree is just one out of a whole set of possible network properties that one can view to identify functionally important residues. So degree is one which is connected to a large number, or a highest degree node would correspond to one which is connected to the highest number of nodes. You could also talk about nodes which are important because of the central position they occupy in a network, or they could be part of a group of nodes which are highly connected to each other. To use a social analogy, if you think of this as individuals and the links corresponds to the social interactions of their acquaintance relations, a highest degree node would correspond to the most popular person in that population, has lots of acquaintances. But oftentimes, the person having the highest number of acquaintances may not be the most important person in terms of linking the network together. That could be, for example, these two individuals who are linking two groups who are otherwise completely separate from each other. So the only way that information can go from this group of individual to this group of individual is if they go through these two individuals. So they are so-called gatekeepers of the network. Okay? So two or more separate parts of the network could be connected by such gatekeepers, and this essentially contributes to their importance. On the other hand, you could have a group of people, each of whom don't have too many connections, but they form an extremely tight clique. Okay? And we all know that tight cliques who all decide to vote together or voice their opinions together can often have inordinate influence on a population. And so therein lies the importance of nodes which are part of a high order core. Okay? So depending upon what you're interested in, you could look at each or every one of these network properties and try to identify residues in this particular case, or it could be any system that you're interested in. You could be using any of these properties to identify functionally important nodes in a system using these properties. So that was about the lowest scale, the protein viewed as a molecular network. Let's go up one step higher. So let's look at intracellular networks. So intracellular networks, there are a huge number of examples. I will just deal with a few. One of them is the protein-protein interaction network. So I mentioned that proteins, when they fold up, form this very unique three-dimensional conformation. Okay? And they react with others by essentially binding together in a kind of a lock and key fashion. And so here is a cartoon of how such, two such proteins bind together essentially to create a protein complex which will result in reactions. Now, if you have a situation where you have more than two proteins binding with each other to form a complex, and that of course is fairly common, you can have a situation of not just pairwise relation between nodes, but a much more higher order interaction. Okay? And this brings us to the concept of the hypergraphs. So in this set of lectures, I'll be mostly focusing on networks. We'll rarely talk about hypergraphs. But it is important that you encounter this generalization of networks, because at least I believe that they are the next frontier. Okay? So what are hypergraphs? So I mentioned that networks are defined by a set of vertices and a set of nodes which link a set of vertices or nodes and a set of edges which link these vertices. But you could easily think of a situation where it's not just pairwise interactions, but multipartite interactions which link nodes with each other. So for example, a protein complex could be made out of 4 different proteins which are bound to each other. Now you could say that, well, what if I represent them as several pairwise interactions? Or in other words, suppose I have protein A, protein B, and protein C like this. I could think of it like this. Okay? Or if it's like 4 proteins, I could think of a structure like this. Okay? But in the context of protein-protein interaction, this leads to an ambiguity. It doesn't tell you whether you are representing the occurrence of separate protein pairs. So, you could have the possibility of AB occurring together. You could have the possibility of AC occurring together. You could have the possibility of AD occurring together. So, that could be what you're representing. Or you could be representing the fact that although individually these pairs are not really functional, once all 4 bind together, you have a functional protein complex, which is this ABCD. Okay? If it's the latter interpretation that you mean, it may not be very meaningful to talk about this complex in terms of separate dyadic relations between AB, AC, AD, and so on. You are better off representing it as a set which consists of the 4 elements A, B, C, D. Okay. So here we see that the concept of network or graph is no longer adequate, and you have to represent a relation between more than 2 elements at a time. Okay. A hypergraph does precisely that. Okay. A hypergraph is essentially defined when you are talking about, again, a set of nodes, n nodes. So that part is exactly the same as the definition of graphs or networks. But now, instead of edges or links, you have hyper edges. And hyper edges are not just talking about a pair of nodes that this particular edge connects, but 2 or more nodes that particular hyper edge connects. Again, I can take a social analogy. So you can think of a situation where instead of focusing on pairwise relations, we are interested to understand how a social network is clustered into groups. Okay? So there could be, for example, members who belong to more than one group. Okay? So you could have individual A, B, C belonging to one group, and then B, C, D could be belonging to another group, and C, D, E could be belonging to another group yet. Okay? So you realize that it's inadequate to kind of express it simply as a pair of dyadic relations of A, B, B, C, C, D, because it does not give you any of this information about in what context they are actually acquainted with each other. They could be acquainted with each other because they're part of a club. They could be acquainted with each other because they are part of some kind of a Facebook group, or they could be acquainted with each other because they are cousins. Okay? Each of these form a very separate cluster, and you would like to represent them using a hyperedge. So in such a situation, you would rather prefer to go into such a kind of description. Okay? So if you think, so going back to the example I'm talking about, if you just think of this as follows, that ACD is part of a particular group, and then they're also part of other groups, and this is how the overall association between them is defined. This defines a hypergraph, and occasionally what we do is we represent it in terms of what is known as a bipartite network. So what's a bipartite network? It essentially tells you about the relation between a set of nodes through the clusters or groupings that they are part of. So for example, over here, you'd realize that A, C, D are part of one group, and so this group I call the green cluster. So I basically connect A, C, and D to a node which corresponds to the green cluster. B and D both belong to the brown cluster. So here I take node B and node D and connect them to another node which corresponds to the brown cluster, and similarly to this mauve cluster. Okay? So a bipartite network is one where I have now, instead of just one type of nodes, two separate classes of nodes with very different interpretations. So one set of nodes correspond to the actual individuals, the other set of nodes correspond to clusters. Okay? And links exist only between one set of nodes to the other set of nodes, but not within the same set of nodes. Okay? So essentially you say that A, C, D are related to each other only via the connection that they have with the corresponding cluster that they belong to. Now you might say that this looks very abstract. Okay? Is there some way in which you can make it much more meaningful? Well, just think of chemical reactions, right? So for example, A, C, D could be all different kinds of reactants. Okay? So one could be, for example, the input, another could be the catalyst, the other could be output. So what relates them together is simply that they are part of the same reaction. Okay? So in this way, one set of nodes would be the reactions, and the other set of nodes could be the reactants. Okay? Now, there is, of course, a way of translating this to the simple kind of networks that we have seen. So you could just do a projection and say that, look, if A, C, and D are all part of the same cluster, I will just connect A with C, C with D, A with E, and so on. Okay? So in other words, I essentially do a projection and get a graph which looks like this. And that's often what is done, because most of the techniques that are associated with the most commonly used software can only handle such simple networks. But what I'd like to point out is that you should be aware that when you're doing this projection, you're necessarily doing a simplification and would be losing possibly very vital information. So it's okay if you do the projection, but you should be aware that you are doing an approximation, that you're doing a simplification, and you might be missing some very important information. Going back to the protein-protein interaction network, so if you do a simple-minded projection where you say that I will only consider dyadic interactions, so even complexes of 4 proteins and so on are just represented as set of several dyadic interactions, you end up with a network which looks like this. So this is for yeast. So there are 1,870 nodes which are connected by 2,240 links. So these interactions were determined by the yeast-2 hybrid method. So this corresponds to the largest connected cluster. So what's the largest connected cluster? So oftentimes when we talk about networks, what we leave unsaid is whether when we connect all these n nodes, they're all part of one large cluster or not. Okay? So it could well be that although I'm talking about a network of n nodes, they could be just fragments which are disconnected from each other. So this could be, for example, one network. Okay? So here the largest connected component is this one, which has the largest number of nodes. Okay? So oftentimes, since there is very little possibility that any perturbation or any information which originates here would come over here, we'd be only dealing with the properties of the largest connected component. Fortunately, it turns out that in most systems which are fragmented, typically the largest connected component occupies the largest share of all the nodes concerned, and the other fragments are essentially microscopic fragments. So similarly here, the largest connected cluster of the protein-protein interaction network essentially accounts for 78% of all the proteins. So hopefully, just by looking at it, and neglecting the others, we would get a rough idea of how the protein-protein interactions of the yeast give rise to the different properties. So one of the things you can immediately do is try to identify the number of connections of each of these nodes and try to find out, do the most connected nodes have certain properties? And what you find is that if you color the nodes according to what kind of effect they have on yeast if they are mutated or removed, you notice that the lethal nodes, in other words, nodes whose removal would lead to immediate death of the organism, typically are associated with the highest degree nodes. In other words, those proteins which connect or which interact with the most number of other proteins also happen to be extremely important for the survival of the organism. So if you remove them, typically it leads to death of the organism. So this association between the likelihood that if you remove the protein, it would lead to a lethal effect correlates extremely well with the number of interactions that a particular protein has in the protein-protein interaction network. So again, this reinforces the fact that even simple-minded network analysis can actually give you important biological insights into a particular network. So I mentioned that the intracellular domain has a plethora of networks. So this is another of those. So this is the network which connects the different metabolites into, to each other through metabolic reactions. So this is basically a cartoon of all the metabolic pathways which are involved in the mammalian cell. Now notice that this is also a simplified picture, because if you really want to be very accurate, you want to represent it in terms of a tripartite network where you have one set of nodes which correspond to metabolites, one set of nodes which correspond to enzyme, and one set of nodes which correspond to reaction. But typically people would just do a projection onto metabolites and see this network. Yet another network in the same domain is the network of genes which are regulating the activation of other genes. So this is the so-called transcription regulation network. So this is work that got the biologist Jacques Monod and François Jacob the 1965 Nobel Prize. And so they figured out that even though each of our cells, each of the many cells in our body have exactly the same DNA, the fact that some cells become liver cells and some cells become kidney cells and some cells become muscle cells is because they express different proteins. So how is it that the cells know which proteins to express? Why is it that they don't express all the proteins? And so they did this wonderful work in which they figured that the reason why a particular cell expresses only a subset of all the proteins it's capable of expressing is because genes essentially influence each other's activity. So a gene typically expresses a particular protein, and this particular protein can often go and prevent the expression of another gene, or maybe it would promote the expression of another gene. So you can view this as a network of switches. So when you put a switch on, that is, you make the gene express a protein, it results in some other switches being put off or some other switches being put on. So it's not like if you focus on a particular gene and look at its expression, it would convey a lot of information. You have to see it in totality. You have to see it how the expression of a particular gene affects the expression of other genes or the inexpression of other genes. And this essentially describes a dynamical system whose different attractors correspond to the different cell types. So this is work that was done by Stuart Kaufman in 1969 and carried over to much more interesting insights about how different cells occur and so on. To cut a long story short, you can view the gene transcription factor network as essentially a bipartite network of genes expressing proteins and the protein in turns causing other genes to switch on and off. And so you can project such a bipartite network onto a simple network and you can get a network of genes which regulate each other. And so you can see that just in the intracellular domain, you could have multiple networks, a network that defines metabolism, a network of protein-protein interaction, and a network of genes, many of whose members are common across the different networks. Many of whose members are influencing directly the members of the other networks. And so, here I've just described a subset of all the possible such networks that occur in a cell. I've already explained protein-protein interaction network, metabolic network, and the gene transcription regulatory network. You also have genetic interaction networks, which essentially tells you that mutation in two genes can often result in a phenotype which is completely unexpected from the mutation of the individual genes. So essentially, they point towards the interaction between those two genes. So this is related to the concept of epistasis. And in a second, I'll be talking about cell signaling. So what this tells us is that while we can view each of these networks in isolation and study its property, if we really want to understand how a cell behaves, we have to now look at a network of networks. Okay? And this brings us to what is in some sense an extremely hot topic in today's network science, the concept of multiplex and interdependent networks. So often they are used interchangeably. Multiplex and interdependent networks mean different things. So multiplex network simply means that the same node could be part of different networks which essentially correspond to different kinds of interaction. Again, let's use a social analogy. When I'm at home, I'm part of my family network. When I'm at the institute, I'm part of a academic official network. And when I'm, let's say, with my friends, I'm part of a friends network. So the node is the same, namely me, but depending upon what context we're talking about, I'm actually part of 3 different networks. Okay? Now, several of these nodes which are there in my academic official network could also be my neighbors in my friends network. Okay? So you can see that there could be some very intricate relations between the different networks of which I'm part of, and that's what multiplex network studies. So it basically looks at that if you have a perturbation which starts at one network, how does it translate across the overall system by essentially traveling across from one network to another network using the links which are there between nodes in these different networks. Okay? So often properties which hold true for a single network would become extremely modulated or become very different if you look at the multiplex network. On the other, interdependent networks talk about two different networks, but which essentially directly influence each other. An example in point is the electrical grid and the network of computers. So typically, a network of computers would be controlling the electrical grid, and the electrical grid would, of course, supplying power to the network of computers. So if now something happens to the electrical grid, it will obviously have an impact on the network of computers. Now that the moment the network of computers start going down, that will have a further effect on the electrical grid. So even parts of the electrical grid which were not affected by the original perturbation in the grid now start going down, which in turn will have, again, an effect of the network of computers. So you can see that they can have extremely complex feedback processes occurring between these two separate networks. So that's essentially an interdependent network. And the thing is that within the cell, all these networks can actually exist in both multiplex relation as well as interdependent relation. I mentioned signaling network very briefly. So this is basically the sequence of reactions that allow a cell to recognize inputs which are coming through signaling molecules attaching to its receptors on its surface and then giving appropriate actions. So this could be, for example, instructions to duplicate itself or cell division, or it could be to kill itself, so apoptosis and so on. And so this is a kind of an extremely complex network which has often been dubbed the integrated circuit of the biological cell. And in a sense, it's like a nervous system, except that here we are not talking about neurons connected with synapses, but rather molecules connected through chemical reactions. And a wonderful example of how this functions exactly like a nervous system is if you look at E. coli, which is a single cell organism, there's no concept of nervous system because you just have a single cell you're talking about, right? But it can do extremely complex tasks like chemotaxis. So if you put it in an environment where you have, for example, food at one end, it can sense the chemical gradient, which is essentially caused by the molecules from the food, essentially diffusing from the source outward. And it can sense this gradient and gradually start moving towards this attractant. Okay? Now, if this was a multicellular organism, we would have tried to figure out, okay, how does this nervous system, allow it to figure out the gradient. In fact, for organisms like C. elegans, that's precisely what we do. People actually study chemotaxis in C. elegans and try to understand how its nervous system is able to figure out its gradient and so on. But in the case of E. coli, there's no neuron, right? So one wonders, how is E. coli able to do this? And then one realizes that E. coli manages to do this just with molecules interacting with each other. So in a very real sense, it's actually functioning exactly as the nervous system will do in the case of a higher eukaryotic organism. Okay, so this is just more details about how exactly the kind of processes happen. So you have a signal which binds to the receptor, which essentially results in a series of chemical changes. But you can formally represent it as a kind of a network. So this is a response, B-cell response signaling network, which is involved in the immune system, whose breakdown leads to disease. And often you can also have pathogenic agents hijacking certain parts of this signaling network in order to make multiple copies of themselves. And so what we're interested here is to understand how is it that such a network is sensitive to different kinds of inputs, and yet it's strong enough to withstand the huge amount of noise that the cell is subject to on a day-to-day basis in the body. And in fact, if that looks complicated, it's actually part of a much larger network which looks like this. So oftentimes when looking at such systems, you can be pardoned to think that signal transduction is a bit like a Rube Goldberg contraption. So Rube Goldberg, if you haven't encountered his work before, was a cartoonist, I think late 19th, early 20th century, whose work consisted primarily of such ludicrous devices where he, for example, would design a self-operating napkin. So here the way it operates is that the person takes some soup on a spoon, and as he is about to put in his mouth, the pull that he exerts on the spoon causes a sequence of actions which involves, for example, a toast popping up and a bird flying off a perch, which involves eventually a rocket flying off with a sickle cutting a string, which would eventually cause a napkin to wipe his face. Okay? Looks extremely complex and fragile, right? But the difference between this and this is that this is remarkably robust. So what is it that gives rise to the robustness of networks? And we come back to this question of why did nature opt for networks where everything is essentially connected with everything else, rather than something as simple as this, where you just have the input elements over here, the output elements over here, and there is almost no interaction between them. Now, if you think about it, this you might superficially say, okay, look, if you think of these elements as threshold elements, and if you are like trying to think, okay, what do they implement? What kind of input-output functions do they implement? Then if you have just a few connections, then there's only so many logic gates that you can implement. Okay? On the other hand, if you start having interactions between them, you could implement things like OR gate, AND gate, and in this particular case, things like exclusive OR, okay? Which are really, really complex logics. But the price that you pay for this flexibility, for this plethora of logic functions you can implement, is that now you need extremely complex control mechanisms. Why? Well, suppose you think of this as a part of a network. So now if you have activation of this node, and if you don't put any checks, signals will just go on and on to the entire network. So the signals will diffuse without control, and you could well have apoptosis instead of cell division, or the reverse in a cell. Okay? So you have to figure out some way that signals are only received by a subset of the network and not everyone who is connected to the original receptor. Now, one way to do that is to just introduce inhibition, negative links. Okay? Another way to do that is to go modular or introduce compartments. Okay? Now, modular networks, which are essentially defined by networks being formed into compartments where within each compartment you have a very dense set of connections and the compartments are relatively isolated from each other, is a very common feature seen in most biological networks. And since they occur at an intermediate scale of the network where they are neither properties of 2 or 3 nodes, neither are they property of the entire network, so that's why we call them a mesoscopic principle. Okay? Something which is only observable at the intermediate scale. Now, you see them right from the intracellular level. So this is the metabolic network of E. coli where all the modules are pointed out, all the way to the ecological networks where the nodes are species, and you can see that there are clearly 2 compartments where within each compartment you have very strong interactions. Between 2 compartments you have very weak interactions. So what we find is that oftentimes the modules which are defined structurally also happen to be associated with specific functions. And one of the open questions is, how are the function and the structure related? And this brings us to the next level, which is the intracellular network. And of course, the classic example is the network of neurons. And a poster child for this is the C. elegans, the Synohrabditis elegans neural network, whose entire nervous system has been mapped out through a series of studies done back in the 60s and the 70s. And so what one finds is that the neural network has a very clearly defined modular organization, which one can relate to the functional circuits, where the functional circuits are related to the various kinds of functions like touch sensitivity, thermotaxis, chemosensation, and so on, which the nervous system is supposed to perform. And one can do analysis at the level of modules which clearly identify these functionally important neurons purely through terms of structural means. So this is one way in which you can take pure structural analysis of a network and point out functional importance of specific nodes. Finally, we go over to the scale of individuals or network of individuals. So this is something that Gautam would cover in detail. So this is the network along which the severe acute respiratory syndrome disease spread in Taiwan in 2003. So the circles are individuals and the diamonds correspond to the hospitals which these individuals went to. And so one of the things you immediately note is that the hospitals served as the conduits through which the infection spread to other individuals. You could also view infectious diseases at a much larger scale, namely that of airline transportation network. So you can see how SARS spread from originating in Hong Kong all the way to the rest of the world. And by looking at how exactly airline transportation network are essentially functioning as high-speed transmission lines for contagia, you can actually say something about the pattern of such disease spreads. And finally, at the larger scale, you can view wave of ecological interactions as a network. And here the interesting questions are, by looking at a network like this, which is the Scotian shelf food web, which is a marine ecosystem, you can try to ask questions like, if you have a sudden decline in the population of this species, how will it affect the rest of the species? Will the ecosystem collapse? Will it trigger a cascade of extinction events? What would be the effect of human intervention on this? And more generally, is higher diversity good or bad for the stability of the network? So finally, as a research program, you can think of the following, that essentially we are focusing on network structure, network function, and network dynamics to ask, how does functional criteria, for example, stability of network dynamics, constrain network structure? So this is the kind of questions that we'll be dealing with in the rest of the lectures. So I conclude with a set of introductory papers and books that you might be interested in looking at. So if you want just one textbook, this I think is the book to go to, Mark Newman's Networks and Introduction. There was he originally published in 2010. It's just come out with a second edition this year. There's a book dedicated to just dynamical processes and complex networks like epidemic, synchronization, and so on, which is slightly dated 2008, but still very good. And of course, there's this wonderful book by Yuri Alon, which focuses just on intracellular networks. So if that's what you're interested in, this is definitely a book you'd like to look at. Okay? And there's a series of papers and popular books that you might want to also peruse. Okay, so with that, I end today's lecture. And so from next lecture onwards, we'd essentially put some more flesh on the skeleton that I outlined today.